Okay, so far in the process of protein synthesis, we've talked about its importance and how they consider it the central dogma of all molecular genetics. And that is because everything in our body is basically made up of proteins. So to recap, before we move on to that final stage that I told you about called translation, let's just try and go back and put everything back into perspective here. So DNA carries the genetic blueprint for everything in the cell that the body needs. Its instructions always remain in the nucleus, and when the cell needs new proteins or any new cell parts, it makes a copy of its original DNA. The DNA is like a reference library with books or materials that you're not allowed to borrow or sign out, but you can just make copies of them. So remember I talked about the importance of the DNA and how it's not necessarily safe for it to leave the nucleus into the cytoplasm because lots of things can happen that might make it non-functional. It might damage it in a way there where it cannot perform the job that it's meant to do. So in order for that to happen, we need to make a copy called messenger RNA. That messenger RNA contains the information that we need, and we can send that piece out into the cytoplasm. So the process of creating this message is called transcription. And now that brings us into the last part. We want to take that message and translate that into what protein we need. So over here, it just sort of recaps. Replication we talked about. How do we just make a strict copy of our DNA? And that would be for making new cells. Okay, we need, in order to take a cell and make 2 new cells, we need 2 copies of that DNA. And then transcription is where we just left off. We still take our original DNA, but because we don't want this guy going out into the cytoplasm, we need to make a copy of it so that that can go out into the cytoplasm, and if anything happens to it, it's still your original DNA is intact. Now we're on to the final process. We call this process translation. So remember how we made a copy of the DNA, but it was in the form of RNA. And the main difference between RNA and DNA is if we break down that genetic code, RNA has the only difference from here to here is that DNA has the letters A, T, C, and G, and each of these has a complement to itself. So A pairs with T, T pairs with A, C pairs with G, and G pairs with C. But when we're making our copy of RNA, all of that still stays the same, except where you need a T, it becomes a U. So thymine becomes uracil. Now we need to take that new code. So this is obviously a code that's different than what we started with, and we have to come up with a protein out of this. So that's what the process of translation is. And in order to understand translation, we're talking about codes. And I left you with that one activity where we're just decoding general sentences based on information in a chart. Well, this is exactly what translation is like. We have this important chart, which we call the genetic code. I believe it's on page 240 in your text, I think. I will verify that for you, but we're going to provide you with a copy of the genetic code anyway. I will put it right in the content for you. Because you can't go through the process of translation without this important chart we call the genetic code. So the sequence of bases or letters on the transcript that we made contains information needed to synthesize proteins. And it's this chart, the genetic code, that helps determine what protein is which. So they are read in groups of 3. Okay, so along our mRNA transcript, every third letter or group of 3 represents something we call a codon. And just like the word code, it codes for a specific protein. And remember, we have 20 different proteins, so there has to be enough codes that tell us those different proteins. So each codon will specifically code for an amino acid, and only one codon will serve as the start. So when we're translating our message off of our mRNA transcript, so we need to know where to start. And so there's something called a start codon that will allow, when we're reading this mRNA, to know where to begin on that gene. Remember, either just segments of DNA that code for certain things. So coded in there will be something called a start codon for each gene, for each protein. And that begins the process of translation. So translation will begin here. And we're going to look at this in detail. So I'm just trying to give you an overview so you can understand once we get there. As I said earlier, there's a start codon, but there's also a stop codon. So there's a code within that set of letters that tells it to start. There's also a different code that tells it to stop, to signal the end of protein synthesis. Okay, so let's have a look at what this genetic code looks like. Okay, so yeah, just like I said, in your textbook, page 240, I will make a PDF copy of this that you can print out and have beside you, because I think sometimes working through these, the process of translation, where you're asked to actually translate an mRNA, you're going to want a paper copy of this. So I will put a PDF copy of the genetic code so you can print it out and have it in your notes. So here's what the genetic code looks like. Depending on which textbook you look in or which website you're looking at, they may have set it up a little bit differently, but the purpose behind it is the same, and the codes will always be the same no matter which one you're looking at. So this is a universal table that's used all over the world, and it's used in the same way. And so what you have to focus in on here is just the order. This is it. So along this side represents, remember I told you that we have 3 letters. So for every 3 letters on your, let me just give you an example while we look at this. You're going to get some practice doing this, but this will make a little bit more sense if we do this. I'm going to move this off to the side. Okay, let's say we had an mRNA transcript. And I'll just use some for example here. Let's just say we had a start codon. So this is part of our mRNA. This is what was made as a copy of our mRNA transcript. So now it could be anything. Oops, and we can't have thymine. If this is mRNA, remember anywhere a T is going to happen, we need to have a U instead. So I'm just making this up, but so you can see how it works. But somewhere along our mRNA, so let's just say this is our mRNA that was copied from our DNA inside the nucleus. We're out in the cytoplasm. We want to make a protein out of this. And remember I said it's going to read along our mRNA transcript and it's going to find what we call a start codon. And you're going to know this is a start codon because they've highlighted your starts and your stops right in your genetic code chart. So the letters that represent our start codon are always A, U, and G. And so remember I said if we have our mRNA, that each set of 3 bases represents a protein, or sorry, an amino acid. And we determine what that amino acid is by using this genetic code. So our start is AUG, and if you didn't even know that, you would just need to know how to come to the genetic code chart and know how to use it. So here's how you use it. There's always 3 codons or bases that make up your codon. The first base starts on this side. So our first one is A. We want to start here, A. Our second letter is U. So the second base is we're using the strip across the top. So our first base is A, second base is U, so we're in this column, and our third base is along this side. So we have to choose which of the 4 within that box. So we're looking at G. So A, U is up here, and G is the 4th one down within that box. So that tells us that AUG represents methionine. Okay, and that would be the first set of codons. And then the second set of codons, or bases, CAU, if we do the same thing, we're starting on the side with C. Second base is A, so it's where those 2 meet. So we're in this box now. C and A meet here. And the third is U. U is at the top, so it's telling us it's histidine. So that's the amino acid that would represent this set of bases. And it just continues along in triplets of 3, or in bases of 3. CGA, so we start with C along the side and G at the top. They both meet here in this box. And our third codon, our third base is A. So C and G, and then A is down here, so it would represent arginine. Okay? So that's basically how the genetic code works. And we're going to look at this in a lot more detail, and I will walk you through the entire process from start to finish on how to do this. I just wanted to introduce the genetic code to you so you understood how things were translated, because we can't understand the whole process of translation unless we know how things are translated in the first place. So this genetic code, or this chart, is universal. It's used by everyone across the world to represent how to take an mRNA transcript and translate it into our specific proteins. Okay?